So let's look into the cell membrane, specifically how it's made and how it functions to keep things in and out of the cell appropriately. So first, let's look at the structure of the membrane, because once again, with everything in biology, we have to know the structure in order to understand the function. So here are all of your key terms for this section. Now, the cell membrane, also called the plasma membrane, is the boundary of the cell that separates it from the outside environment. It's incredibly thin, only about 8 nanometers, and the main function of the cell membrane is to control what goes in and out of the cell. It keeps the cell safe from anything that's toxic, it brings in food, it removes waste, so it's really important in many, many ways. And you can see down here at the bottom, that black line is an actual image of the 2 layers of our plasma membrane from an electron microscope. So our cell membrane has 4 main characteristics. It is fluid and flexible, and by fluid we just mean it is all the pieces move around easily. We're not talking that it's a liquid. Number 2 is that it's selectively permeable. This means it chooses what comes in and out, and it blocks what it wants to come, or what it does not want to come in and out. 3, this same structure of the double membrane that we're going to talk about, the double layer membrane, that is going to be the same structure for not just our plasma membrane, but also for all of our membrane-bound organelles. And 4, it's made up of lipids with some proteins and carbohydrates for added extra functions. So the membrane, let's start with number 4 there, is made up of a chemical that we call a phospholipid. So this is 2 hydrocarbon tails, so just carbon and hydrogen repeated over and over. And we have 2 of those tails with what we call a phosphate head. This is a polar molecule up at the top there. The phosphate is negatively charged, making the head of the phospholipid going to have a partially negative charge. The tails, on the other hand, are neutral. They are nonpolar, so they are hydrophobic. The head is the only hydrophilic part. So this makes this molecule what we call amphipathic. It has a polar part, but it also has a non-polar part. Amphi means both, pathos means experience. So this is a molecule that is both a polar and a non-polar molecule at the same time. Now, because of the structure of the phospholipid, that directly forms the overall structure of our membrane. So if our head of our phospholipids is hydrophilic and the tails are hydrophobic, and the inside of our cells, that cytoplasm, is mostly water, and the outside of our cells is mostly water, so that means that the tails are going to try to hide away from the inside and the outside of the cell. And the easiest way to do that is to make what we call a phospholipid bilayer. There are 2 layers of phospholipids facing in opposite directions. So you have one phospholipid with the tails pointing down, and on the other side you have another phospholipid with the tails pointing up, and they face each other. This allows those polar heads, those hydrophilic phosphate heads, to face the water. So they're facing the water because they're polar, and for these non-polar parts to be blocked away from any water. So they stay further away and they're not interacting with it at all. So you can see here in this image, you see a bunch of those phospholipid heads on the outside. And then on the inside, we get this nice non-polar area where all of the tails congregate. And that's going to be creating this barrier that does not really allow anything that likes water, anything that's hydrophilic, to pass through. So, as I said, the cytoplasm and the extracellular fluid are basically water. So the hydrophilic heads face the water, and this creates a barrier that it's a little harder for water to get through, and anything that is dissolved in water can't get through the membrane without help. This is what allows the cell membrane in particular to control what goes in and out. Let's look at the characteristic that says that the membrane is fluid and constantly changing. So the phospholipids are actually constantly moving around each other, partially just because, remember, all molecules are in constant motion just because of inherent characteristics, and they get more motion with more heat and less motion with less heat applied. Now, this movement of these phospholipids allows the entire membrane structure to appear as if it is fluid, and it really is fluid in the sense that we mean it's changeable, it's constantly moving around, it's constantly adjusting itself, it's not static or remaining the same. And because of this movement, it actually does make it easier for things to get in and out of the cell, because the movement allows certain molecules to pass through as these phospholipids are moving around each other. If they stop moving, they would harden, and it would make it a lot more difficult for things to pass through. Now, some phospholipids have double bonds in their hydrocarbons, so they have an unsaturated fat in their fatty acid tails, and that creates a kind of kink shape in one of their tails. And that actually forces all the phospholipids around it to be pushed away to create some of that space and allowing that movement. So when you have an organism that is high in unsaturated fatty acids, that is contributing directly to the fluidity of their cell membranes. But now, some organisms don't have a lot of unsaturated fatty acids in their membrane. Instead, they're only going to have relatively straight fatty acids in their phospholipids. And when that happens, those tails force those molecules to pack very, very close together. And this makes the membrane more viscous or more solid almost. And that prevents important movement within the membrane to allow for objects to pass in and out easily. So often, organisms that are highly saturated, meaning the tails are close together versus the unsaturated tails that have the kinks, when it's very saturated, they will actually put molecules of cholesterol in between the phospholipids to force that space and to force that fluidity. So the whole point of cholesterol is to increase the space between our phospholipids when all of their tails are saturated fatty acids. The unsaturated fatty acids, those produce space completely on their own. So cholesterol increases the fluidity of the membrane. Now, the next characteristic that we're going to discuss about the membrane is its selective permeability. So selective means you're choosing, and permeability means the ability for something to pass through it. So this is saying that the membrane chooses what it allows in and what it does not. So some molecules can pass through, but not others. So let's look at which molecules can move across, which ones cannot, and why, because that's something you have to be able to understand. So let's look at lipids. Anything that is hydrophobic, like a lipid, do we think it can pass through or not? Well, let's look at our membrane itself. So we have the hydrophilic heads on the outside and the hydrophobic tails, that nonpolar area, on the inside. These are all, honestly, lipids right here. So we have to look at this inner area and decide, can something move across that highly lipid area? So if I'm already a hydrophobic lipid, it makes sense that I would be able to pass through this hydrophobic area without a problem. And that actually is what happens. So lipids can pass right through because they can go through that nonpolar area without disrupting those hydrophobic tails. What about something very hydrophilic like a salt? Could that pass through this area here? Well, salts are basically, not basically, they are charged ions dissolved in water. That's most often how we find them, especially in living things like a cell. So if we're talking about a highly charged, highly hydrophilic substance that's dissolved in water, it has to bring water with it as it moves. And this is a very, very hydrophobic area that does not want any water to pass through it. So salts cannot pass directly across the membrane. What about polar molecules? Well, polar molecules are also hydrophilic, and so for the same reason, they are not going to be able to pass through. Nonpolar molecules are hydrophobic, so yeah, those ones should be able to pass through. And once again, things like sugars, those are hydrophilic molecules, just like our salt example. So things that are hydrophilic or polar cannot pass through. Things that are hydrophobic or nonpolar can pass through. That's the basic idea here. There's a few other stipulations, however. If it's polar, it cannot cross. If it's nonpolar, it can, because we have to say, can it pass through this highly nonpolar area? And if it's nonpolar, it can. Now, large molecules pose a different threat to the membrane. If you try to take a gigantic molecule and move it through the membrane here, that's going to really disrupt the structural integrity of the membrane and potentially cause a really weak point in the cell, and the cell can end up lysing or bursting if we bring too many large molecules in like that. So large molecules cannot directly pass across the membrane. We have ways of getting them in, but often not in their intact form. Now, really, really small molecules actually can pass through, also depending on their charge and whether they're polar or nonpolar. So small molecules usually will be able to squeeze through the membrane there as long as they are pretty nonpolar. Now, gases almost always can pass through. Things like oxygen and carbon dioxide and nitrogen. They are so small, and for the most part, they are very nonpolar. So those things, like oxygen gas or CO2 waste, that can just go directly across the membrane. Now we have small polar molecules. Yes, those can, because they're small enough. So this is where we kind of get a caveat. Yes, it's polar, but if it's small enough, then it can pass through. Any polar molecule that's a little bit larger, though, cannot. And by small, we mean incredibly tiny. It has to be, very small. Large polar molecules cannot, because they are polar, and they wouldn't be able to tolerate that non-polar area of the hydrophobic tails, and also because of their size. So this area in the middle with all of our hydrophobic tails is impermeable to large and polar molecules. So we call this movement across the membrane diffusion. So let's look at some examples of what can and cannot diffuse across the membrane. So what can? Fats and other lipids, because they're nonpolar. Small uncharged molecules like our gases, oxygen and CO2. Very, very, very small polar molecules with no overall charge. If it's an ion, it has an overall charge, and that's a very different situation. Remember, polar molecules overall are still neutral. However, when we talk about the slightly positive and the slightly negative areas, those can cause problems down the road. So if it's really small and polar, it can make its way through. If it's a normal to large polar molecule, it cannot. So what cannot get directly through the membrane? Anything that is very polar and a normal to large size, something like glucose, sugar, any charged ions like salts, and any large molecules like starches and proteins. So how do we get some of these things in the cell? Because glucose, that's our food, right? That's what cells eat. How do we get the glucose into our cells to be able to get energy from it if it's polar and it can't cross the membrane? We need proteins to build up amino acids for ourselves often. So where do we get those proteins? Where do we get, the food into our cells overall if it's one of these molecules that cannot pass through? Well, that's where we bring in membrane proteins. We often call these transport proteins or um, membrane channel proteins, but we can just call them membrane proteins. That's probably the easiest way. And their job is often to transport polar or charged molecules across the membrane. And these proteins are often very specific. They'll transport just, positively charged ions, or just glucose, or just some other protein. So we get very specific channels for these larger or polar molecules. And you think about it, it's a stable hole inside the membrane that allows water, that cytoplasm and that extracellular matrix to pass through. So this stays polar the entire time. It never has a non-polar moment inside the channel as the molecules are moving through. Now, this is also often anchored into place, so these can move around within the membrane, but they're very much anchored to the membrane itself with our cytoskeleton or with other types of molecules that are kind of binding it within the membrane itself. So these proteins have a lot of different unique jobs. The main one is that transport of materials across the membrane, but we have tons of other membrane proteins that can receive signals, they can send out signals, they can cause one cell to stick to another. They can act as enzymes that are performing chemical reactions and functions at the level of the cell membrane itself. They can hold antigens, those identity name tag markers that go on every single cell. So our membrane proteins have many, many different jobs. So you can see here a bunch of those different job examples, like the transporter, an enzyme. We'll talk about signal transduction a little bit later. It's basically receiving a signal on the outside of the membrane, and then this protein causes that signal to get sent in a different way on the inside of the membrane. This one here is holding up an antigen. This is an example of two cells being pulled together and adhering together, and here's an example of how it attaches to the cytoskeleton. We also have carbohydrates in our membrane, and those are going to be, the example in this image is the green hexagons. Now our carbohydrates are always going to be on the outer facing side. They're never on the inside of the cell membrane, always on the outside. If you think about it, carbohydrates are our sugars, so always think that cells are candy coated. They only go on the outside of that membrane. Now, what do we use these for? Mainly to recognize other cells and for communication. So really, these are the antigens for our cells. Oftentimes they can have other functions, but these are the two main ones. Now let's look into the physiology of the membrane. So we looked at the anatomy, what is the structure. Now we're looking at the physiology, the function. Here are a few of your terms. Some of them we'll discuss now, some of them we'll discuss in class. So when we talk about how things cross the membrane, we have to understand the second law of thermodynamics. And that is that the universe is trending towards disorder. It's entropy. Entropy is a measure of disorder or chaos in the molecules of the universe. So molecules have a natural tendency to move from an ordered state to a disordered state, where everything is equal. Now, living beings don't like that. We like things to be very ordered, and we need them to be in order to do many different chemical reactions and to get energy and things like that. So really, when we think about the second law of thermodynamics, we can think about it in the way that these two graphics are showing us here. So on the top one, what is the likelihood that if we put two gases inside a chamber that are relatively the same weight, obviously, what is the odds that they would naturally form a barrier where all of one type are on the top and all of another type are on the bottom? That is incredibly rare. What most likely happens is that they just randomly mix until the amount of each type is equal throughout the entire container. Also, if you have a handful of bricks, if you were to ever throw those bricks out, what is the probability that they create a wall? Incredibly unlikely. That is a very ordered state that requires work or energy input to get there. What most likely happens is they just fall in a pile of randomness. So think of entropy as randomness and disorder. So to understand this on the level that we need to for the membrane, we have to understand concentration gradients. So a concentration, I've used this term a couple times, but let's formally define it. A concentration is just an amount of a material. So if I say there's a high concentration of sugar in my tea, that means there's a high amount of sugar in my tea. Now, a concentration gradient is in existence when particles of one type are congregated in one area versus others. So we can see it in this image right up at the top here, where all of these blue particles tend to congregate on the right side, and there's very, very few of them on the left side here. So now we say that going towards where all the molecules are is going up the gradient or against the concentration gradient. When we're going from the area where all the molecules are down to where there's few, we call that down the gradient or with the gradient. Now, in the world, gradients are artificial. Whether they are made by cells or by humans doing things, if we have a gradient, it is a temporary state that is most likely created by a living organism. So this gradient will only be temporary because all of these molecules that are gathered together on that high end, they don't want to stay there. They want to move down the gradient, so from this high amount to the low amount, until they reach what is called equilibrium. This is a state of balance where the particles are equal everywhere in the given space, or where they're equal on both sides of a membrane. That's going to be what we talk about for most of today. So the gradient is where there's an imbalance in the amount of the material from one area to another. Equilibrium is what basically the second law of thermodynamics is saying. The universe is trending towards equilibrium, and living things don't like equilibrium. That kind of is antithetical to us being able to efficiently do work and use the least amount of energy to do that work. So looking more closely now into this idea of moving with or against our concentration gradient, let's connect it back to the cell. So when cells are moving materials across their membrane or just across the cell in general, they do so either using passive transport or active transport. Passive transport is when the molecules just diffuse naturally on their own, following that second law of thermodynamics, going from order to disorder. So this is going from a high concentration to a low concentration until everything is equal. This is passive, meaning there is no input of energy here. This just happens naturally due to the normal movement of the molecules following that second law of thermodynamics. The other one is active transport. This is attempting to create more order out of the disorder. This is going against the second law of thermodynamics. As a result, if you were trying to fight the natural tendencies of the universe, you have to pay. And what are we paying with? We're paying with energy. So you got to pay the energy tax in order to create more order rather than more chaos. So in this case, when organisms want to move materials against the concentration gradient from an area of a low amount and push it towards where there's a high amount, that is not what nature wants to have happen. So it requires energy in the form of ATP often. There's often other types of energy that the cell uses too, but the one that is most commonly used and the one that we're going to discuss the most is that use of ATP. Now, diffusion, I've used this term a couple times, is a form of passive transport. Diffusion is just the movement of particles from a high to a low concentration until equilibrium is reached. That's what we call diffusion. So think of diffusion as this example here, where if you put a couple drops of food coloring into some water, eventually that food coloring will diffuse out and the molecules will reach an equilibrium where there's equal amounts of blue dye throughout the entire glass of water. Think about spraying Febreze in your room. You spray it in one area, but the particles get carried throughout the whole room until the whole room smells nice. Now, in our cells, each material is going to diffuse down its own concentration gradient. What do I mean by that? If I have a bunch of salt and a bunch of sugar and a bunch of some lipid, each of those does not care about the other. If there is more sodium outside the cell and less sodium inside the cell, it will move from the outside to the inside until it's equal. Even if the sugar amount is already equal, even if there's more sugar inside than outside, everything will move down its own concentration gradient. There is a slight caveat to that. Sometimes it will depend on the charge, because if a molecule is very positive, even if it wants to diffuse across the membrane, if the membrane on the inside already has tons of positive charge, it might not be able to diffuse as easily, but that really gets into diffusion of water. And that's a whole other case scenario. Water tends to break all the rules. So just think of, for the most part, most substances will diffuse on their own gradient. So they will go from an area of a high amount to an area of a low amount until they are equal with that equilibrium on both sides. So let's look at an example here. If I have 6 molecules of a lipid on the outside of my cell, or the inside, I'm sorry, and 8 molecules of a lipid on the outside of my cell, I have the high concentration on the outside and the low concentration on the inside. So which way will these lipids move across the membrane? Will they move inside the cell? Will they move outside the cell? And why? Why do they do that kind of movement, and what is that process called? Well, they're going to move from the outside to the inside. You can see that little arrow here that came up. Because I have a higher concentration on the outside of my cell, and it needs to move to where it's a lower concentration until we reach equilibrium. That process is called diffusion. And why does it happen? Well, because we're just following the second law of thermodynamics here. Now, what about for molecules that can't directly diffuse across the membrane? What if it's a polar molecule or a large molecule or a charged molecule? What do we do then? Well, then you have something called facilitated diffusion. To facilitate is to help. So in this case, you're just diffusing the molecule still, but with a little bit of help from a channel protein, one of those membrane proteins. So it's still passive transport. There's no energy involved because we're just following the second law of thermodynamics. We're moving from a high amount to a low amount. But since these can't directly cross the membrane, they need the help of a carrier protein to do that. And often these are very specific. They'll only open once a certain concentration difference is met, or they'll only open once a certain voltage in the cell membrane is met. There's a lot of different caveats placed on when these channels open up or not. Now, active transport is going against the flow. Rather than letting it go from a high amount to a low amount, we actually want to go from a low amount to a high amount. And so this is going to require the direct input of energy, often in the form of a molecule called ATP that's made by our mitochondria. So now the cell actually does this all the time. It will create concentration gradients so that it can use that stored potential energy, kind of like a dam, open up that dam when it needs energy, and then the flowing of the molecules down their concentration gradient is used to either spin a motor within our cell, a protein motor, or just to capture that energy and use it somewhere else in the cell. We're going to see this example in cellular respiration with how we make energy, ATP, out of our food. So we create these concentration gradients frequently in our cells. Active transport also allows us to have bulk transport. So sometimes we need to bring in very, very large molecules. Sometimes we need to bring a lot of molecules in all at once, or the opposite. Sometimes we need to get rid of a lot of junk all at once. So to do that, we use a bulk active transport, one of two types, either exocytosis or endocytosis. Exocytosis, think exo sounds like exit. So this is releasing bulk materials from the cell. So a little vesicle, remember a vesicle is just a tiny bubble of membrane, is going to contain some kind of waste or some type of signal molecule or some type of bulk material that we want to remove from the cell. So what happens is that vesicle will move towards the membrane and then fuse with the membrane itself. When it does that, it dumps all of its contents into the extracellular environment, the outside environment. So we most often use this for secretion, where we're releasing a signal out into the greater body or out into the environment if you're just a little cell that lives that way. Endocytosis, think endo sounds like into. This is bringing large or bulk materials into the cell. So our white blood cells will do this when they do something called phagocytosis, when they want to eat a bacteria or they want to eat a cell that has been contaminated by a virus or something like that. So they will actually wrap their membrane out and around that bacteria and form the vesicle around it and bring it into the cell. Often then what will happen is that vesicle will fuse with a lysosome. Remember, lysosomes are like the stomach of the cell. And it will dump all of that acid and those hydrogen peroxide and that, those digestive enzymes into that lysosome, into that vesicle that just took in all the food, like the bacteria, and it'll digest it inside that vesicle. So there's a couple different forms of endocytosis. I mentioned the phagocytosis, where we're reaching out the membrane to bring in a food particle. You can see a real life example of that in that GIF there. There's also penocytosis, where the membrane just dips in and brings in liquid and whatever happens to be in the liquid. It's not looking for anything in particular. And then we also have receptor-mediated endocytosis. Here's where we have little receptors on the surface of our cell. And when the correct molecule binds to that receptor, it causes the membrane to start to dip inward to create that vesicle and bring in that specific molecule that just binded to those receptors. The last thing we're going to talk about is osmosis. This is just a special case of diffusion. It's passive transport or diffusion of water. That's all that it is. We just use a special name because it's water, and water tends to, break all the rules. So water can actually diffuse straight across the membrane without needing to use any proteins, even though water is polar. And that's because water is very, very small, and it's so essential for life that we kind of evolved around our ability to get water in and out. So water can diffuse directly across. Or we have these really cool proteins called aquaporins. That literally means like water hole. So these are special channel proteins that allow water to move quickly in and out of the cell. They're not found in every single cell of the body. They tend to be found more in places like the kidneys, where we're filtering out a lot of blood, which has a high water content, and other types of cells like that. Now, as I said, osmosis is just the diffusion of water across a semi-permeable membrane. So across our membrane, this is osmosis right here, diffusion across the membrane. But when water diffuses, when osmosis happens, it moves based on what all the other molecules are doing. So it looks at how much stuff is dissolved in the water and then looks at where the water needs to go to equal itself out relative to how much stuff is dissolved inside of it. So let's look at an example here. Here we have a permeable membrane, a semi-permeable membrane with water on both sides. But on the right side, we also have sucrose or sugar. Now, which way is the water going to move? If you look at it, the water level is equal on both sides of this membrane. So originally you might think, well, no diffusion is going to occur because it's already at equilibrium. However, that's not true. There's a lower percentage of water content on the right-hand side because some of that space is being occupied by those sugar molecules. So the water is actually going to move towards the right in an attempt to reach equilibrium. And so if you actually do this with the correct semi-permeable membrane, you will see that the water level will actually be different on either sides of the membrane. So let's look at a little bit more into how we can predict the way the water is going to move. Animal cells and cells that are part of multicellular organisms, they like to be in a state that we call an isotonic solution. They like to be isotonic. In this case, it's technically an environment where the water levels are at equilibrium on both sides of the membrane. This means there's going to be no net movement of water across the membrane. Now, the wording of that is key. There's no net movement. Net means overall movement. There's still going to be water moving back and forth across the membrane because molecules don't stop moving unless they're at absolute zero, and that's a bad state. So there's constant movement of these molecules, and as a result, some are going to slip out of the cell, some are going to slip into the cell. It's just natural. But overall, they're going to move in and out at equal rates. So if two molecules come in, two are going to move out to maintain that equilibrium. It's kind of like if you have, here's equilibrium, it's basically going to be an oscillation right around that equilibrium point, where sometimes you're going to have too much inside the cell, sometimes too little, but overall you're at equilibrium here. So as a result, the cell is going to stay the same size. It's not going to get bigger by bringing in too much water. It's not going to get smaller by losing water. Now, like I said, since these molecules of water are constantly moving, remember, diffusion never stops. It just reaches a steady back and forth where it's equal back and forth. This is how our cells like to be. So our cells like to be isotonic. That's a way you can remember that term. I like to be, or I am isotonic. I like to be isotonic. You also have hypertonic solutions. So hyper means above, and this term tonic or tonicity refers to water pressure. So this is above water pressure. So what we mean here is that there's more solute in the solution than in the cell. Okay, that's a little confusing. What do we mean by that? There's hyper, more above the amount of stuff dissolved in the water outside of the cell than is dissolved in the water inside the cell. So think about taking one of our cells and sticking it in the ocean. There's much more dissolved salt in salt water than there is inside the water of our own cells. So we're going to have, the way I like to think about it is, let's draw a little beaker here. Let's pretend there's a cell inside. And let's say that this cell is 99% water, and that the water outside is only 97% water. The rest is sodium and chlorine because we're putting it into the ocean, let's say. So there's more other stuff inside or outside of the cell, inside the beaker, than there is in my cytoplasm. So when this happens, the way that the water is going to want to move is from high to low until we get to equilibrium, because this is just diffusion. So the cell will actually lose water to its environment. So the net movement of water is going to go out of the cell. So the water is going to leave the cell, and as a result, the cell is going to shrink. And this can be really bad for the cell depending on how much water it loses. When we're talking about an animal cell, we call that crenated. Basically, it just means it starts to shrivel up. When it's a plant cell, we call it plasmolysis. This is where the cell membrane starts to pull away from the cell wall of a plant. But either way, this is not a good scenario. Cells do not like to be hypertonic. Nothing likes to be hypertonic. I'm going to write that up here. Nothing likes to be hypertonic because water is so important, and we don't want anything to steal our water if we still need it. The other solution is what we call a hypotonic solution. This is one where there's a lower concentration of solutes or dissolved materials outside of the cell than inside of the cell. So let's draw my handy dandy little beaker and cell here again. So let's say that this cell is 98% water, 2% stuff, and we drop it into 100% distilled water. Now there's more water outside compared to inside, so that water is going to move into the cell from the environment. As a result, there's going to be a net movement of water into the cell. The cell will take on water and it will grow or it will swell. Now, sometimes it will take on so much water that it will do something called lyse. You might have also heard the term lysis. And this is where the cell bursts and dies. So our animal cells do not like this state. We do not want to be taking on water because we don't have a cell wall or anything like that in place to protect us from all that pressure of the water moving in. However, bacteria and plants like to be in this state. They want to have constant water pressure moving into the cell so that they are in a state of kind of swelling, I guess you could say, and that pushes their membrane out against their cell wall and makes it easier for them to survive in the environment where finding water is a little bit more of a problem. So here we can see some examples. We have the hypotonic solution on the left, and this is where the animal cell may lyse, but the plant cell and the bacterial cells are happy. Then you have the isotonic solution in the middle. This is I am isotonic. This is where our animal cells like to be. And then the dreaded hypertonic. Now I know it's kind of silly, but I was told a way to remember this when I first started teaching, and it's made it so I've never forgotten. If you think about these terms, you can say hypotonic is OMG, I'm growing, and make like a big O shape with your arms like a cell taking on lots of water. I am isotonic, and hypertonic, I'm shrinking. I'm shrinking, OMG, I'm growing. So, like I said, kind of weird, but I promise you it'll help you keep those straight. And that is it. We'll go into some more examples of real-world diffusion and passive and active transport, and we'll discuss rates of diffusion a little bit more in class.